Oh, come on, Sam. Let's think about it. Think about the logistics. These cis-regulatory mutations are bound to be far more important in evolution.
Benito, this is... So everyone knows structural mutations are far more important.
There's no evidence. You're talking absolutely bollocks.
So much evidence.
Oh, exon smexon.
Well, at least I'm no sissy.
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 Exon smexon.
Well, at least I'm
no sissy.
Exon smexon.
Well, at least I'm no sissy. Well, at least I'm no sissy. Well, at least I'm no sissy. Well, at least I'm no sissy.
Fair enough. Hello, it's the 8th video in the Evolutionary Ecology series. God, we're speeding through them, aren't we? Now, last time we talked about the short-sighted watchmaker and some of the limits that there are to adaptation and evolution, okay? Which is something we hadn't really considered before. And what we're going to do is basically carry on from where we left off. Now, at the end of the last video, we started to consider whether evolution was predictable, particularly at the genetic level, and we sort of talked about the divided field in evolutionary biology on what types of genes are involved, okay? So cis-regulatory and structural genes. And we're going to talk about them in more detail today. A good example of a structural mutation is something that we've already seen before in these Florida beach mice. This was studied by Hoekstra, remember, one of those people who thinks that structural mutations are more important for evolution. And he identified the MC1R gene that we've talked about, which is associated with coat colour. And remember that we talked about that this MC1R gene isn't just present in mice, it's arisen multiple times in all sorts of different animal taxa. So although different mutations occur independently across the species range, they all seem to be occurring in the same genomic regions. So this is hinting that perhaps there's some constraint to the amount of genetic evolution that's possible, and hence making it more predictable. So therefore, these possible adaptive exon changes, so mutations encoding DNA, are limited to a small region of the genome. And that's likely to be due to what we were talking about before, these pleiotropic effects. If you mutate one locus, it may be beneficial for some aspect of the trait that locus produces, but it may be deleterious in others. Now, a model animal, which we haven't talked about at all in this series yet, but which is quite surprising really, because it's used loads in these kind of studies, and that's Drosophila melanogaster, a fruit fly, okay? And these show differences in pigment expression, which affects the camouflage and the temperature regulation of the fly. And this is due to differences in ebony expression, so a gene called ebony. And what we see is that the mutation that creates these differences is fairly downstream in the pathway of ebony production. So that means if it's further downstream in the pathway, that means it's less likely to affect other traits. Okay, there's going to be less pleiotropy going on there. So that seems to be hinting at the idea that there is some constraint. It's called an enhancer locus where that mutation is. Okay, so perhaps over time these mutations have been refined to this small region, this enhancer locus. So to reduce the amount of deleterious effects elsewhere, possibly in other tissues. Okay. There's another interesting mutation called the shaven baby mutation. And this mutation results in the loss of larval bristles in Drosophila. And the mutation that causes this is in between two cascading molecular pathways. And because of this, it has many pleiotropic effects. It affects other traits. And it's been found that differences in this gene, whether this mutation is present or not, is what creates most of the differences we see between Drosophila species because of all these pleiotropic effects. So maybe it's these kind of genes that we need to look at when talking about things like speciation, because a mutation here can have multiple effects. Now, when looking at different levels of variation in Drosophila species, we begin to see differences in the mutations that arise within and between species. So higher in the taxonomic scale results in genes which affect modules, okay, so genes which affect loads of other different genes, such as this shaven baby mutation. But within species, we see more structural mutations. Okay, so this takes us right back to the end of the last video about these cis-regulatory and structural mutations. Which one's more important? This seems to suggest that in short-term evolution, these structural mutations are more important, but in long-term evolution, when we're looking and comparing between species, these cis-regulatory mutations are more important. And this is exactly what's been studied by Stern et al. In 2009. We see contrasts in genomic architecture when looking at short-term and long-term evolutionary processes. Now, in domesticated species, such as cattle or whatever, we do see more structural mutations, but that's because of the amount of husbandry provided by humans and a lot of the ecological pressures on domesticated species have been removed. As humans have been domesticating our plants and animals over the years, we've been more specific, if you like, on what traits we want to change. Okay, so therefore we've only selected for those structural mutations. Now, due to these limits in the mutational landscape that we've talked about, we can begin to see that some mutations are reused during evolution. And this is something we've already talked about before in the third video when talking about the Prestin gene. Yeah? The Prestin gene has been independently selected in bats and dolphins for its use in echolocation. So therefore, these bats and dolphins share morphological features that have been independently derived. Okay? Which is amazing considering, bats and dolphins occupy very different niches, don't they? So this is what we call genomic convergence. This could indicate that there are very few places in the genome where a mutation for echolocation could occur. Okay? So there's a shortage in the amount of genes which selection can act upon to affect echolocation. Right? That could be a possibility. Or, over time that we've been talking about, the amount of mutations that affect echolocation has been refined over time because of these negative pleiotropic effects. Perhaps bats and dolphins started off with very different genomic architectures, with mutations that possibly had deleterious effects, but maybe these were offset by these bats and dolphins being able to hunt prey effectively, having evolved echolocation. And over time, these mutations have been refined to a proportion of the genome where these deleterious pleiotropic effects are minimal. So this seems to suggest the opposite to what we were saying. Evolution may actually be quite free. And we can study this by looking at what proportion of echolocation efficiency is controlled by shared genes rather than non-shared genes. Now, we've talked about the adaptive landscape before and how it's an excellent framework for studying evolutionary ecology. So basically we have suboptimal peaks which are separated pretty much by valleys. However, real adaptive landscapes are probably likely not to be in 2 dimensions like in that diagram I showed you in the second video, I think it was. They're likely to be multi-dimensional. And a good way of studying this is by looking at Müllerian mimicry. Now, what's Müllerian mimicry? So Müllerian mimicry is where you have got one toxic animal or unpalatable species mimicking the morphological characteristics of another unpalatable species. So the key thing is that they're both unpalatable. If one wasn't, then that would be called Batesian mimicry, okay? Just so we get our terminology right. And this is seen in Heliconius butterflies, and I'm about to show you absolutely astounding pictures, absolutely incredible. So have a look at this. So all the butterflies on the top row are more closely related to each other than the 3 butterflies on the bottom row. How amazing is that? Okay, so these two, we can call them 2 separate taxonomic groups, have appeared to show incredible parallel evolution. Right? So, how on earth did these butterflies get it exactly right? Okay? It's pretty astounding. So here we see divergence within the taxa and convergence at the same time. This creates multiple mimicry rings with strong positive frequency-dependent selection. Now, Nadu et al. In 2011 wants to look into this further. He wants to look into the genes involved, and he found, yes, the same genes were involved in these colour patterning phenotypes. There's therefore predictability then, isn't there? The genetic evolution, and this is the case for divergent and convergent phenotypes. But how did these mutations arise so quickly? Surely the chances of looking exactly like another species is incredibly small. Okay? Well, recent evidence is suggesting that these colour pattern genes may have spread by hybridization. This allows these mutations to get established more quickly in the genome, because it would be very rare otherwise. In fact, it would be so rare they'd be pretty useless mimics. But despite these constraints, clearly mimicry is an incredibly effective strategy to avoid getting eaten. Okay? So, evolution therefore depends on genetic variation in fitness. We've known that right from the first video. And throughout this video, we've been wanting to know what proportion of all that variation is useful for adaptation. Now we know the bigger the effect a mutation has on fitness, the quicker it will spread, the stronger natural selection will act, yeah? But remember, if we want to talk about quantitative genetics, the heritability of a trait is almost as important. Remember, in the first video we talked about those 2 things that natural selection needs, and that's variation and heritability. Okay, so it's no use for a trait just to be under genetic control. It needs to be inherited, right? That's obvious. So therefore, evolutionary change is limited by heritable variants in fitness and also affected by correlations between traits, which has been a running theme throughout the past 2 videos, pleiotropy. So these are all things that we need to consider when we are talking about portion of all this variation that we see in the genome, what is important to adaptation. And again, we'll look more into that next time. So I'll see you then.